Hello students, welcome to LEELA'S tutorial. So today we'll discuss about inoculation and incubation of explant in plant tissue culture media. So in this topic, the temperature and light regimes play an important role, right? Without their maintenance, you cannot produce callus, right? So let us see first, it is the surface sterilization process. So in last class, we have discussed that it is the explant, that is the plant tissue, any part of the plant tissue which acts as a starting material for your plant tissue culture, right? So that explant has to be selected based on physiological age, size, location and season of availability, right? Once you select the plant, then you're going to select the part of the plant, whether you're going to select the leaf or petals or the shoot fragments, root fragments or meristem, whatever, right? So here I have selected leaf as the explant. So once the selection of explant is done, all the dust and the contaminants present on the surface of the explant must be sterilized, right? Must be cleaned. So here you are going to go for surface sterilization process by using surface sterilants. So first step will be rinsing them under running tap water in order to avoid any dust particles. Then you are going to wash them in distilled water, then transfer them into disinfectant along with detergent, then again rinsing in distilled water, sterilized distilled water, then transfer them into 70% ethanol followed by mercury chloride, right? Either you can make use of mercury chloride or sodium hypochlorite or bromine water or hydrogen peroxide or silver nitrate. Any one of the thing you are going to use. After that you are going to rinse it with double distilled water and then you are going to place them in laminar airflow where the Petri plate is overlaid with stack of filter papers on which your surface sterilized explant is allowed to air dry. So once the explant is air dried, you are transferring that into another sterilized Petri plate and here you are going to select the region or the size of the explant which you have to inoculate into the media. So here with the help of scalpel you are going to excise that and again the excised portion is transferred with the help of the forceps. Right, you are going to use the forceps for transferring your explant. So now you have surface sterilized explant. Already in previous classes we have discussed how to prepare semi-solid media and the liquid media. So here then after preparing your media explant, now next step will be your inoculation. Right, so here I have shown you the semi-solid media. Right, it can be the semi-solid media. Why you are calling it as semi-solid media? Because you are making use of only 1%. Right, you are making use of only 1% or 1.5% of agarose in your MS media or Murashik and scoop media. Right, you are going to pour the media either in the plant tissue culture tubes or here the vessels or it can be a Petri plate. Right, so any one of the thing you can pour your media and allow it to set. So now it is a semi-solidified media, semi-solid media where you are going to transfer your explant. With the help of forceps, you are going to transfer them in this plant tissue culture tubes or jars or Petri plates. Right, everything must be done under aseptic conditions. Right, so in the laminar airflow only you are going to perform. So after inoculation, it will be your incubation. So let us see how you are going to inoculate in liquid media. So here the media composition and the components are same in semi-solid culture and also in suspension culture. What is the difference? Only thing is here you are making use of 1% agarose, 1 or 1.5% agarose, whether here there is no agarose added. Right, so the media remains in liquid condition. So simply the explant will be immersed in the liquid media and you are going to culture them. If you are going to culture your explant in semi-solid media, you call it as semi-solid culture. If you are going to culture your explant in liquid media, you call it as liquid cultures or suspension cultures. Right? So once you have done with the inoculation, the next step will be incubation, that is maintaining your explant. Maintaining means you have to allow this explant to stand by or resist to these environmental conditions. Right? It is an in vitro condition. Right? They have to resist to this and get tolerant to the in vitro condition, and also they must start dividing. The cell division must start. For that you have to provide certain conditions. Right? Certain environmental conditions must be provided. So now these jars after inoculation, they must be either transferred to the plant tissue culture racks, otherwise into an incubator where it is automatically all the conditions are maintained in it. Right? So here let us see the temperature. Mostly for the explants we maintain at 25 to 28 degrees centigrade. But some explants they require high amount of humidity where they must be incubated at less than 15 degrees centigrade. Right? So this depends based on the explant you are using. Okay, then the light intensity, mostly it is 1000 lux of light intensity must be provided. And here the light and dark cycles must be shifted for every 12-hour duration. Right? For 12 hours the light must be provided and another 12 hours the darkness must be maintained. Right? So like this you are going to maintain for 15 days. So here in the nutrient media or Murashik and scoop media, you have added with the auxins. So what is the use of auxins? They induce cell division. Right? Or they initiate the cell division. Once they get standby within 15 days, now see before inoculating this explant, you are going to otherwise you can see here. Here this is the leaf. The lower epidermis of the leaf is ruptured gently without tearing the explant. So that lower epidermis which is ruptured gently will be placed over the media. So that means the epidermis or the cuticle which is present will not obstruct the intake or absorption of the nutrients. Right? So here you make easy absorption of the nutrients. So now the nutrient media also consists of auxins. So now these auxins which are absorbed by this explant, the cells present in the explant, what happens? The cell division is initiated. Right? So the cell division which is induced by auxins, as a result they start dividing. So they start dividing and within 3 to 4 weeks you can see a ball like structure or a mass of cells which are formed. Right? This is nothing but your callus. Right? How you define the callus? Callus is an unorganized mass of cells. These are undifferentiated. Undifferentiated means the cells which doesn't have any structure or function. Is that clear? So from the differentiated explant, here the leaf is having structure and also the function, photosynthesis. Right? From this differentiated explant, you have transformed it into a callus which is undifferentiated mass of cells. Now these mass of cells can be micropropagated to produce any part of the plant or a complete plant. Right? So let us see how we are going to induce the callus in detail in next class.